Hplc utilizes a liquid mobile phase to separate the components of a mixture. Analytes are first dissolved in a solvent and then forced through the column under high pressure. Hplc is very versatile because of its ability to easily separate a wide variety of chemical mixture, which include non-volatile and thermally unstable compounds. For example, organic, inorganic, biological samples, synthetic or natural polymer, and thermolabile compounds. We have 4 advantages of Hplc. First, greater sensitivity, various detector. Second, reusable column, many analyzed. Third, ideal for ionic species and large molecules. And the last one, sample recovery, fraction collectors. Hplc application, any compound with solubility in a liquid that can be used as mobile phase, analytical or preparative, highly purified compound in a small or large quantities. Application include analysis of sugars, pesticide, residue, organic acid, lipid, amino acid, toxin, and vitamin. Principles. First, begins by injecting the solute analytes onto the top of the column. Second, separation occurs as the analytes and mobile phase are pumped through the column. Third, each compound eludes from the column is applied on the recorder. And the last one, the response of the detector to each component is displayed and known as chromatography.
The objective is to determine the retention time of a standard solution, to identify the caffeine peak in a soft drink sample, and to determine the amount of caffeine in soft drink sample using response factor method.
Now, let's move on to the methodology. Firstly, the preparation of caffeine standard solution. First, solution A, the stock solution with 10 mg per ml. Firstly, weight 10 mg caffeine into a 10 ml volumetric flask, and then dissolve in distilled water and fill in more distilled water up to the calibration mark. Second solution, which is solution B, with the working solution of 0.1 mg per ml. Firstly, pipette 1 ml of solution A into a 10 ml volumetric flask, and then fill in with distilled water until the calibration mark. Next, the sample preparation. First, pour 10 ml of soft drink sample, which is Coke, Pepsi, and 100 plus, into a small beaker. Second, use a magnetic stirrer to remove any carbonation. And third, filter 2 to 5 ml aliquot through a 0.45 micromolar pore diameter membrane filter to remove particulate matter. And last but not least, the injection and analysis. First, inject 10 microliter solution B into the Hplc to establish the retention time of caffeine. Second, measure the peak area of caffeine. Third, inject 10 microliter of the prepared soft drink sample into the Hplc instrument. And last but not least, measure the peak area corresponding to the caffeine by referring to its retention time. Now, let's get to know the function of each component in the Hplc equipment. Firstly, the mobile phase is for the organic aqueous solvent that are used depending on the analysis. Next, the degassing unit is to remove the dissolved air from the mobile phase. Next, the solvent delivery pump works to deliver the mobile phase at constant flow. The injector works to introduce the standard solution or real sample to the Hplc column. Next, the sample vial is to store standard solution or sample solution. Column works to separate each compound contained in the sample. The column oven is to keep the temperature constant. Then, the detector works to detect the eluted compounds from the column. And last but not least, the signal from the detector is processed and the chromatogram is displayed in the workstation.
For the first sample, we use standard caffeine sample. For the retention time is 2.482 minutes, the peak area 6249.48779. For the peak height is 1196.10278, and area percentage is 100%, or it can say as pure caffeine reference. First, we analyze a pure caffeine sample. This chromatogram shows a single sharp peak at a retention time of 2.482 minutes. Since this is a pure caffeine solution, it serves as our reference. The peaks at area height represent the amount of caffeine present. We will compare this with other samples to identify the caffeine content. For the RF caffeine standard is 262494.8779. Next is Coke sample. For the retention time for Coke sample is 2.481 minutes. The peak area is 7001.11365. Peak height is 1465.59729. And the caffeine area percentage is 57.36%. And other peaks present suggest additional compounds in Coke. Next, we analyze the caffeine content in Coke. The chromatogram shows a peak at 2.481 minutes, which closely matches the retention time of the standard caffeine sample. This confirms the presence of caffeine in Coke. However, the caffeine peak accounts for only 57.36% of the total area, meaning other compounds are present. These could be sugars, flavoring agents, or other ingredients in the soft drinks. Next is the Levita sample results. For the retention time for caffeine is 2.219 minutes. For the peak area, 10342.1, and the peak height is 1183.27344, and the caffeine area percentage is 16.41%. While the other peaks, strong peaks at 2.010 min and 2.799 min suggest multiple compounds. Now let's look at Levita and energy drink. The caffeine peak appears at 2.219 minutes, slightly earlier than in the standard sample. This could be due to slight difference in the sample composition. However, caffeine only contributes 16.41% of the total detected compounds, which means Levita contains a significant amount of other active ingredients. These could include chlorine, B vitamins, or sweeteners, which are common in energy drinks.
Hplc analysis has given the interpretation concerning retention time and peak area that quantifies the concentration of caffeine in the given sample. Chromatogram inform about separation of compound. Sharper peaks indicate effective separation has taken place, while border peaks denote column overloading. The calculation of the response factor was performed using a standard calibration curve that was further employed in calculating caffeine concentration. The common problem encountered, such as peak broadening, overlapping peaks, and baseline noise, were resolved by adjustment in the flow rate, mobile phase, composition, and calibration of the detector. While minor, this challenge proved that the Hplc technique is applicable for identification and quantification at high accuracy and therefore important in analytical chemistry. By that, we know that the Hplc analysis accurately quantified caffeine concentration by interpreting chromatographic peaks, applying the response factor, and addressing minor issues like peak broadening and baseline noise, demonstrating its reliability in analytical chemistry.
For the conclusion, this experiment effectively utilized Hplc to detect and quantify caffeine in soft drinks by identifying its retention time and applying the response factor method for precise measurement. The findings confirm caffeine's presence with differences in peak area and height indicating varying concentration, while other peaks suggested additional compound. Minor issues like peak broadening and noise were resolved through adjustment, demonstrating Hplc as a repeatable technique for caffeine analysis in beverage.